होमवर्क कैसे पूरा होगा? चलो स्कूल जाओ। मम्मी आज छोड़ दो, कल से हम स्कूल जाएंगे। जाओ नहीं तो तुम्हारे पापा को बुला दूंगी। नहीं नहीं, पापा को मत बुलाओ, हम स्कूल जा रहे हैं। चल चिंकिया। ए ऊपर वाले, इतना बारिश बरसा कि गांव में बाढ़ आ जाए और हमारा स्कूल भी डूब जाए। तुम्हारे कहने से बारिश थोड़ी ही आ जाएगी। सुना चिंटू, बादल गरजने लगा, अब जरूर बारिश आएगा। अरे हाँ रे चिंकिया, मैं तो बारिश में खूब नहाऊंगा, बहुत मजा आएगा। बारिश आने लगा, बारिश आने लगा, ये अब हम स्कूल नहीं जाएंगे, ये अरे चिंकी जल्दी घर चलो, देखो बारिश बहुत तेज आने लगी है, हमारे किताब भीग जाएंगे। हाँ जल्दी घर चलो, आज नदी पर मछली मारने चलेंगे चिंटू। रुक चिंकी, ये देख, ये मछली अभी आसमान से गिरी। पकड़ लो इसे हम भूज कर खाएंगे। मर गया बचाओ, इसे जल्दी खींच चिंकिया, इसने मेरा नाक काट लिया। कूदना बंद करोगे तब तो उसे पकड़ूंगी। हाँ जल्दी खींचो रे दादा, खींचो रे, बहुत तेज काट रही है। ठीक है खींचती हूँ। जल्दी इसे फेंक दो और घर चलो। देखो गांव में कितना पानी भर गया है, लगता है अब गांव में बाढ़ आने वाला है। चलो अरे तुम दोनों इतनी जल्दी चले आए? मम्मी बाहर बहुत तेज बारिश हो रही है और लगता है गांव में बाढ़ आ गया है। जरा मैं भी तो देखूं बाहर। अरे तुम तो सही कह रहे थे, गांव में बाढ़ आ गया है। मम्मी अगर गांव डूब गया तो हम कहाँ जाएंगे? पता नहीं चिंकिया, अभी हमारा प्यारा सा घर भी पानी में बह जाएगा। अरे मम्मी वो देखो, एक छोटा कुत्ता पानी में डूब रहा है। अरे चिंकिया, तू पानी में क्यों कूद गई? जल्दी वापस घर में आओ, नहीं तो डूब जाओगी। नहीं, मैं उस कुत्ते के बच्चे को बचाऊंगी, वो डूब रहा है। मैं आ रही हूँ कुत्ते के बच्चे तुम्हें बचाने के लिए। ध्यान से चिंकी, डूबना मत। मम्मी उधर भी देखो, एक बकरी और गाय भी पानी में बह रहे हैं। डरो मत, मैं तुम्हें कुछ नहीं करूंगी। अरे मैं तो तुम्हें बचाने के has a sequence of bases in DNA that codes for the synthesis of one polypeptide, and genes must somehow transmit their information from the nucleus to the cytoplasm, where protein synthesis occurs. The synthesis of an RNA molecule from DNA is called transcription, and the formation of a protein from RNA at the ribosome is called translation. Kinds of RNA. Each of the three major kinds of RNA has a specific role in protein synthesis and is produced in the nucleus from DNA. Messenger RNA is a linear strand that carries a set of genetic instructions for synthesis protein into the cytoplasm. Transfer RNA picks up an amino acid in the cytoplasm, carries them to ribosomes, and helps position them for incorporation into a polypeptide. Ribosomal RNA, along with proteins, makes up ribosomes. The genetic code. DNA must code for the 20 different amino acids found in all organisms. The information carrying capabilities of DNA reside in the sequence of nitrogenous bases. The genetic code is a sequence of 3 bases. A triple code shows the genetic code as reflected in the messenger RNA that will be produced from DNA. Each 3 base combinations is a codon. More than one codon can specify the same amino acids because there are 64 possible codons, but only 20 amino acids. Their characteristics of the code is referred to as a degeneracy. Note that not all codons code for an amino acid. The base sequences UAA, UAG, and UGA are all stop signals that indicate where polypeptide synthesis should end. The base sequence AUG code for the amino acid methionine, which is a start signal. Transcription. The genetic information in DNA is not translated directly into proteins but is first transcribed into messenger RNA. Transcription involves numerous enzymes that unwind a region of a DNA molecule, initiate and end messenger RNA synthesis, and modify the messenger RNA after transcription is complete. Unlike DNA replication, only one or a few genes are exposed and only one of 2 DNA strands is transcribed. One of the important enzymes of this process is RNA polymerase. After a section of DNA is unwound, RNA polymerase reorganizes a specific sequence of DNA nucleotides. RNA polymerase attached and begin joining ribosome nucleotides, which are complementary to the 3' ends of the DNA strand. In RNA, the same complementary bases in DNA are paired, except that in RNA, the base uracil replaces the base thymine as a complement to adenine. Newly transcribed messenger RNA, called the primary transcript, must be modified before leaving the nucleus to carry out protein synthesis. Some base sequences in newly transcribed messenger RNA do not code for proteins. RNA splicing involves cutting out non-coding regions so that messenger RNA coding region can be read continuously at the ribosome. Translation. Translation is protein synthesis at the ribosomes in the cytoplasm based on the genetic information in the transcribed messenger RNA. And other type of RNA called transfer RNA is important in the translation process. It brings the different amino acids coded for by the messenger RNA into a ligand so that a polypeptide can be made. Complementary pairing of bases across the molecule. Maintains transfer RNA configuration, the process of some unusual bases, thymine, cytosine, guanine, and adenine, or uracil, disrupts the normal base pairing and forms loops in the molecules. The center loop has a sequence of 3 unpaired bases called the anticodon. During translation, pairing of messenger RNA codon with its complementary anticodon of transfer RNA appropriately positions the amino acids that transfer RNA carries. Ribosomes, the sites of protein synthesis, consist of large and small subunits that organize the pairing between the codon and the anticodon. Several sites on the ribosomes are binding sites for messenger RNA and transfer RNA. At the initiation of translation, messenger RNA binds to a small separate ribosomal subunits. Attachment of the messenger RNA requires that the initiation codon AUG of messenger RNA be aligned with the peptidyl site of the ribosome. A transfer RNA with the complementary anticodon for methionine binds to the messenger RNA and a large subunit joins, forming a complete ribosome. Polypeptide formation can now begin. Another site, the amino acidyl site, is next to the P site. A second transfer RNA whose anticodon is complementary to the codon in the A site is positioned. Two transfer RNA molecules with their attached amino acids are now side by side in the P polypeptide and A sites. This step required enzyme aid and energy in the form of guanine triphosphate. An enzyme peptidyl transferase, which is actually a part of the larger ribosomal subunit, breaks the bond between the amino acid and transfer RNA in the P site and catalyzes the formation of a peptide bond between them amino acid and the amino acid in the A site. The messenger RNA stand then move along the ribosome at distance of a codon. The transfer RNA with two amino acids attached to it that was in the A site is now in the P site. The third transfer RNA can now enter the exposed A site. This process continues until the entire messenger RNA has been translated and polypeptide chain has been synthesized. Translation ends when a termination codon is encountered. Protein synthesis often occurs on ribosomes on the surface of the rough endoplasmic reticulum. The positioning of ribosomes on the endoplasmic reticulum allows protein to move into the endoplasmic reticulum as the protein is being synthesized. The protein can then be moved to the Golgi apparatus for packing into a secondary vesicle or a lysosome. Changes in DNA and chromosome. The genetic material of a cell can change, and these changes increase genetic variability and help increase the likelihood of survival in changing environments. These changes include alteration in the base sequence of DNA and changes that alter the structure of number of chromosomes. Point mutation. Genetic material must account for evolutionary change. Point mutation are changes in nucleotide sequence and may result from the replacement, addition, or deletion of nucleotide. Mutations are always random events. They may occur spontaneously as a result of base pairing error during replication, which results in a substitution of one base pair for another. Although certain environmental factors, electromagnetic radiation, and many chemical mutations may change mutation rates, predicting what gene will be affected or what. शुरू करें? What the nature of the change will be is impossible. Some mutations may be unnoticed or even beneficial. However, the consequences of genetic changes are usually negative because they disturb the structure of proteins that are the products of millions of years of evolution. Variation in chromosome number. Changes in chromosome number may involve entire sets of chromosomes, as in the polyploidy, which was discussed earlier. An euploidy, on the other hand, involves the addition or deletion of one or more chromosomes, not entire sets. The addition of one chromosome to the normal 2n chromosome number is a trisomy, and the deletion of a chromosome from the normal 2n chromosome number is a monosomy. Error during meiosis usually cause an euploidy. Non-disjunction occurs when a homologous pair fails to segregate during meiosis first, or when chromatids fail to separate at meiosis second. Gametes produced are either deficient in one chromosome or have an extra chromosome if one of these gametes is involved in fertilization with a normal gamete. The monosomic or trisomic condition results. Aneuploid variations usually result in severe consequences involving mental retardation and sterility. Variation in chromosome structure. Some changes may involve breaks in chromosome. After breaking, pieces of chromosome may be lost, or they may reattach but not necessarily in their original position. The result in a chromosome that may have a different sequence of genes, multiple copies of gene, or missing genes. All of these changes can occur spontaneously. Various environmental agents such as ionizing radiation, certain chemicals can also induce these changes. The effects of changes in chromosome structure may be mild or severe, depending on the amount of genetic material duplicated. Inheritance patterns in animals helps us to predict how traits will be expressed in offspring before these offspring are produced, something that has had profound implications in agriculture and medicine. One of the challenges of modern genetics is to understand the molecular basis for these inheritance patterns. The fruit fly Drosophila melanogaster is a classic tool for studying inheritance pattern. Its utility stems from its ease of handling, short life cycle, and easily recognized characteristics. Studies of any fruit fly trait always make comparisons to a wild type fly. If a fly has a characteristic similar to that found in wild flies, it is said to have the wild type expression of that trait. In the examples that follow, wild type wings lay over the back at rest and extend past the posterior tip of the body, and wild type eyes are red. Numerous mutations from the wild type body form, such as vestigial wings, reduced servile wings, and sepia, dark brown eyes, have been described. Segregation. During gamete formation, genes in each parent are incorporated into separate gametes. During anaphase, first of meiosis, homologous chromosomes move towards opposite poles of the cell, and the resulting gametes have only one member of each chromosome pair. Genes carried on one member of a pair of homologous chromosomes end up in one gamete, and genes carried on the other members are segregated into different gametes. The principle of segregation states that pairs of genes are distributed between gametes during gamete formation. Fertilization results in the random combination of gametes and bring homologous chromosomes together again. A cross of wild-type fruit flies with flies having vestigial wings illustrate the principle of segregation. The flies come from stocks that have been inbred for generations to ensure that they breed true for wild-type wings or vestigial wings. The offspring progeny of these crows have wild-type wings and are the first generations of offspring or the first filial F1 generation. If these flies are allowed to